Hello, everyone. Welcome to join today's talk. My name is Xingtong Liu. I'm a technical information scientist from Jackson Laboratory. For today's topic, which is the mouse models for immuno-oncology, we will basically discuss more about the immunobiology of the hematopoietic stem cell humanized platforms. We do have the PBMC platforms as well, but due to the time limited, I would encourage you to contact me or contact us if you want to know more information about that. And again, we will be discussing more about preclinical tumor efficacy with monoclonal antibodies, bispecific, and CAR-T therapies. And last part, we will be touching base on autoimmunity as well using the hematopoietic stem cell in grafted mouse models. But before we get started, I want to spend a little bit of time to introduce you about the Jackson Laboratory. So Jackson Laboratory is a nonprofit research institution, and we have our own mission, which is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. In order to accomplish these goals, we are working hard towards different aspects. So first of all, we have our own biomedical research labs, and they have their own funding resources to investigate the biology and the disease of the human diseases as well. Partial of the research would be using our mouse models. We also provide preclinical services and resources to researchers all around the world. And lastly, we are also educated to our education outreach. In addition to the talk today you are participating in today, we also sponsor traditional conferences, webinars, seminars on site, and educating programs from different ages. And lastly, through our research, discovery, and education programs, we want to bring innovative new methods and services to the researchers all around the world that who also committed to improve human health. So JAX is a global organization that we are hosting events and shipping our mouse models to researchers all around the world. And you can see right now we have 4 different campuses that either at Bar Harbor, Maine, Farmington, Continental, the Sacramento, California, or we have a newly opened facility right now in China. And also all the shaded areas are the countries that JAX has shipped our product and services to. Okay, let's dive into today's presentation. I want to divide it into 4 different chapters. For the first chapter, we will have an overview of how JAX humanized platforms can provide solutions for preclinical drug lead selection, efficacy, and safety. And for the chapter 2, we will introduce the immunobiology of hematopoietic stem cell in grafted humanized mice. And for the chapter 3 and 4, we will share some applications of the CD34 humanized mice to evaluate the immune modulation, autoimmunity therapy, and also covering the bispecific antibodies, CAR-T, CAR macrophages as well. So let's start from the chapter 1, a very brief overview of the humanized platform that can provide solutions for preclinical drug lead selection, efficacy, and safety. Again, how can we help in this preclinical lead selection? So for a very good lead or target, the basic requirement would be need to be both efficacious, safe, and druggable. And for a monoclonal antibody candidate, what you want to test first is the PK, the pharmacokinetics. You need to understand the half-life of your drugs so that it can help with dosing. And for the monoclonal antibody, which compromise of the Fc portion, and that can be engineered to impact the half-life. And the Fc portion can be bound with the Fc receptor, also called FcRn. We have FcRn platforms that our mouse models has been knocked out of the mouse FcRn receptor and also expressing the human FcRn, and which can accurately measure the half-life of this particular antibody. And again, for efficacy and safety, we have both the CD34 humanized mice or the PBMC humanized mice. As I mentioned before, we will not have time to introduce the PBMC humanized mice, but I do want to mention this is a very good platform for not only testing the drugs for the graft versus host disease, but also it can allow you to testing the cytokine release syndrome, which will be very important if you want to proceed with your candidate. You want to make sure this this potential drug is safe to use in the human beings, is in the clinical settings as well. And CD34 humanized mice, of course, is a very useful platform that can be used to testing the efficacy and safety of your drugs. And we will touch on more details in the next lot of slides. And for the bispecific or trispecific antibodies, which are made through taking the targeting front end of two different antibodies and combining them to create a product that can bind with different targets. And for some of the bispecifics, they still have the Fc portion, but I mean, for this particular one, they are not. So it may or may not feasible for the PK to the FcRn platform to testing the half-life. But they can still be applying to the humanized platforms, either hematopoietic stem cells or bispecific or PBMC humanized mice to test in the efficacy or the cytokine release syndrome within this candidate. In this talk, we will also share some data with you on the CAR-T for immunotherapy, and there are some work done also on CAR-NK, CAR-myeloid cells, or CAR-macrophages. We will have some data on that as well. But I mean, we have experience with working with CAR-T, by which you can put the CAR-T back to the humanized mice, and then you can, with the tumor, and you can test in the efficacy as well as safety. And also, many people may be interested in the gene therapy, and for many disease types, including like not only cancer, HIV, and the immune system related disease, the gene therapy actually offers a very attractive approach by either overexpressing the functional transgene permanently, or gene disruption, gene correction, or specific transgene integration into a precise site, such as endogenous locus of a genome. And technically, you can even consider the CAR-T therapy to be one of the gene therapy by which it will genetically modify the T cells in vitro and you inject them back into the same patient. And for gene therapy, we can also apply in our humanized platform to testing not only the efficacy as well as safety. So that's very brief for chapter 1. And in this second chapter, the chapter 2, let's move on to the human cord blood derived hematopoietic stem cell humanized mice platforms. And here actually is a procedure that we can inject the hematopoietic stem cells into the host mice. We have different variants, and we will touch on more details on the kinetics in the next slides. We will wait for some time until we validated the B cells, the T cells, and more than 25% of human CD45 positive cells in the peripheral blood, and then we can ship out the animals to you, or we can even conduct studies in our facility to testing all the disease-related questions. So the NSG, SGM3, and NSG-IL-15 mice are the 3 strains that we are routinely humanizing them to create a cohort. For NSG mice, after engrafted with the hematopoietic stem cells, they actually have excellent functional human T cell responses. But again, they also showing limited NK and myeloid lineage development in this particular host. And the reason could be not all the lineage of human cells can respond to the mouse growth factors. So we create a NSG SGM3 mice, which expresses the human interleukin 3, CSF2, and kite ligand, and which actually dramatically promote the AML, human AML engraftment efficacy, and also improves the human myeloid cell development in this host. Again, the development of human NK cells would also need the human IL-15. So we created the NSG IL-15 mice, which expresses a physiological level of human IL-15. And this strain also develop a significantly higher level of functional human CD56 positive initial killer cells post-engraftment. I will show you more validation data in the next few slides. And first we did is a side-by-side comparison between the NSG versus SGM3 or NSG versus NSG-IL-15 mice. And these are 2 different studies. On the left side is the first study. We compared the NSG and NSG-SGM3 mice 12 weeks post-engraftment. So what you see here is that the overall engraftment tend to be higher in the SGM3 mice, as you can see the higher percentage of CD45 positive cells, which also leads to more CD4 positive T cells and within the CD3 positive T cell populations. We can also see a much higher levels of the T regulatory cells, and most importantly, we do see a higher engraftment of CD33 positive myeloid cell populations in the SGM3 mice. On the right side is a direct comparison between the NSG versus NSG-IL-15 mice as a host for the hematopoietic stem cell engraftment. So you'll see most of the human immune cell populations are very similar between these two, except for the much higher engraftment of CD56 positive and CD16 positive double positive natural killer cells in the NSG IL-15 mice. And very quickly, here is a long-term engraftment that we compared the NSG mice versus SGM3 mice, and we checked for the T regulatory cells or myeloid cells populations percentage in these slides, and you can see no matter from the percentage or from the exact cell numbers per microliter, from the SGM3 mice, we do see a higher populations of the T regulatory cells and the myeloid cells. You may wonder about why these cells start to increase much higher as for percentage, and they start to decline. And this is because the development of T cells and B cells will turn to be later. And once the T cells and B cells start to expand, you should expect to see the decline of the percentage from the T regulatory cells and the myeloid cell lineages. So if we look at the IL-15 mice as a host for the engraftment, one important question you may always ask is whether these natural killer cells can maintain their immunological features and whether they are functional. And here we did examine the natural killer cells that has been isolated from the peripheral blood of the humanized NSG IL-15 mice. And we do find that these cells, they can express the cell surface marker that either active or respond or suppressive receptors in the natural killer cells, including CD16, KIR2D, different families, KIR2DL3, KIR3DL1, NKG, NKP30, those markers, and which indicating that they are still functional and they can still maintain their immunological features. And what about the function of these natural killer cells in the humanized NSG IL-15 mice? What we found is that the natural killer cells from the humanized IL-15 mice, they do have a similar granzyme B compared with the human peripheral blood. And what the granzyme B is actually one of the effector molecules that can release useful cytotoxic activities. And if you compare the NSG, which in blue, versus the NSG IL-15 mice, you'll see there's a big peak of the granzyme B negative natural killer cells from the NSG, humanized NSG mice, which indicating they are immature. And furthermore, if we take out these natural killer cells for in vitro killing, by which you just co-culture the natural killer cells with the chromium 51 labeled K5062 cells in different ratio. And what you'll see here is the natural killer cells from humanized NSG IL-15 mice do show a similar in vitro killing if you compare it with the natural killer cells isolated from the peripheral blood. And one last point I want to make from this slide is that the NSG IL-15 mice do have more than twofold more natural killer cells per spleen than the NSG mice. So to summarize of this chapter, we're engrafting the CD34 hematopoietic stem cells into the NSG, SGM3, and IL-15 mice on a monthly basis. The mice can develop and maintain stable multi-lineage immune cell engraftment. And certain human immune cell populations require specific growth factor for development and function. And lastly, selection of the platform will be depending on the cell populations and immune mechanism of interest. So next, what about we putting some tumors on board? And the way how this study can be done is actually shows here that first of all, you need to humanize mice with the CD34 hematopoietic stem cells. And then you can onboard, you can inject the tumors into the mice, and they can testing your monoclonal antibodies or your specific therapies in these oncohuman models. So again, always the first question you may wonder is that, can we co-engraft the tumor in a humanized context? And the answer is yes. From our studies, we have done engraft different PDX models into the humanized context, and we do find the PDX models can grow in a different growth rate. And the ratio would be around 85 to 90% of tumors would be able to grow in the humanized mice, no matter of the MHC match status. And in this particular paper published 2020, this researcher groups, they actually took the NSG humanized mice. They put a wide range of the human cancer cell lines derived xenograft into the mice. And what you see here from the left, that those different tumors, they can be engrafted and they will be growing on a very different growth rate, as you can see here. Another observation from the slide is that you will see some tumors, they intrinsically cold that has a very low infiltration of immune cells. And some tumors, such as the HCC827, MB231, they have a high infiltrations. But again, the infiltration of immune cells might not correlate with the growth curve. If you check for the MB231, they have a very high infiltration of CD45 positive cells, but again, they grow quite well in the humanized mice. And also from the same report, this researcher groups actually found that the T cell infiltrations frequency is driven by the tumor, but not by the donor. We have done a similar study in our facility and do show a similar result. From this slide, they use the multiple donors to engraft into the NSG mice, and they put the MB231 cancer cells in the flanks of the mice. And you'll see from different studies, they showed a very similar overall T cell numbers, CD4 or CD8 positive T cells. And this is also true with the rest of the human cancer cell lines that they tested, as shown here, that very similar CD3 positive T cells within the different cancer cell lines that has been tested. So our conclusions from this slide would be the overall quantity of infiltration can vary between the studies, but the frequency of the tumor, the lymphocyte infiltration distribution is similar between different donors. Again, the next question that you may wondering is that can you immune modulate the cells to respond to the tumor? And the answer is still yes. And in this particular study, we have tested the different PD-1 inhibitors. So one is pembrolizumab, another is nivolumab. And those monoclonal antibodies, they can bind to different epitopes in the receptor, and they have very different affinities. So what we found is from the two different donors that the effect of pembrolizumab and nivolumab are very similar, that they can reduce this tumor growth here, which is MB231 in grafted humanized mice. And however, you should also notice that the checkpoint inhibitor responses to varies across different donors. You may see here a little slightly different, but again, it's possible that from some of the donors, they may not responsive. And overall, you may see a 30 to 35% of response rate, and which actually mimics what we see clinically. And you can do other things with tissue models, such as the depletion study. So the GITA is actually expressed in the regulatory T cells, and if you apply an anti-GITA monoclonal antibody, also called MK4166 here in this study, the Fc portion of this antibody will be recognized by the natural killer cells and macrophages. Through the natural killers-driven ADCC, antibody-dependent cellular cytotoxicity, or macrophage-dependent cellular phagocytosis, that you can actually improve the CD4 positive T cells and improve the tumor cell killing. What you can see here from this study, they used the humanized NSG mice and engrafted with SKMEL5 melanoma cancer cell lines. And then these cells were treated with the MK4166 or a control. First of all, you'll see the tumor growth has been greatly inhibited if you treat it with the anti-GITA monoclonal antibody. And furthermore, if you check in for the T regulatory cells within the tumor, you see a depletion of regulatory T cells in the tumor, as well as increased effector cytokines, interleukin-2, interferon gamma within the tumor. And here is an example that we characterize the natural killer cell function in vivo using the humanized NSG L50 mice. We have been tested in 3 different donors, and we engrafted the DALD cells into the humanized mice. The DALD cells will express the CD20 cell surface marker, and they typically resistant to natural killer cell lysis. So if we treated the tumor with the rituximab, which is anti-CD20 monoclonal antibody, you can see that it can induce the natural killer cell mediated ADCC killing, and you can see the tumor is inhibited in different donors, A, B, C. But again, the efficacy is different depending on the different donors you have been used in the study. So what about the bispecific antibodies within the humanized platform? Here we testing the bispecific, also called BITE, that can recognize EGFR on one end and recognize CD3 positive T cells on another end. So if you apply these bispecific antibodies, what we want them to do is to link the T cells and drive the T cells to be targeting the EGFR expressing tumor cells, and so that increases the killing. So what we have done here is that we set up experiments to either a prophylactic trial that we start the treatment when the tumor volume is still small, or we call this therapeutic trial if the tumor has reached a larger tumor size at around 380 cubic millimeters. And after the treatment, we're testing the T cell levels and all the other cell surface markers within the peripheral blood, including CD45 positive T cells, CD8 positive, CD4 positive T cells, and B cells, and nature killer cells as well. For prophylactic trial, we start treatment when the tumor size are still small, and for the therapeutic trial, we start treatment when the tumor reaches a larger tumor size. So you'll see here shows the results that from the prophylactic trial, you do see the BiTE treated group has showing a very dramatic decrease, and the tumor even getting light cleared during the study time. And here on the left side is the mean tumor volume. On the right side is the individual tumors, and you do see that the tumor has been greatly inhibited. From the therapeutic trial, we also see the tumor has been greatly inhibited, but again, if you check the individual tumors, there are some of the individual tumors may not respond that well to the bispecific antibody, and this is as what we expected. So what about the cytokine release profiles within this model? We know that for CAR-T and bispecific antibodies, one of the concerns within these therapies is that once they treated the patient, they may possible to induce a cytokine storm, and they for the as a side effect, and for the one that are severe, it can even threaten human's life or even have some organ failures. So in this particular humanized models, we treated the mice with a bispecifics, and we measure the cytokine release profile 16 hours post engraftment, post the treatment. So you'll see here, no matter from the prophylactic trial or from therapeutic trial, we do see that after the BiTE treatment, we see a great release of the cytokines, including interferon gamma, IL-6, and IL-10. Another notice is that when the tumor size are larger, you do see a much stronger cytokine release if you check for the earlier study days versus late study days. And if you compare the prophylactic trials versus therapeutic trials, you do see a stronger cytokine release from the therapeutic trial compared with the prophylactic trial. So what about CAR-T therapies? It can also use the humanized platforms to testing the therapies within the HSC hematopoietic stem cell engrafted model. In this particular report, the researchers actually used the humanized NSG SGM3 mice, and they isolate the T cells from the SGM3 mice, and they transduce them with the chimeric antigen receptor construct, either anti-B cells or the myeloid cell antigen CD44. And then they put in this CAR-T back into a new cohort of the hematopoietic stem cell engrafted mice. And you can see, first of all, that this CAR-T can mediate specific depletion of the B cells or the monocytes, depending on which antigen it has been expressed using the And during these responses, they also observed the cytokine release associated with the CAR-T therapy, such as the dramatic loss of body weight and the increase of the leukocyte interleukin-6, the cytokine expression levels interleukin-6 has been dramatically increased, as well as the increased body temperatures, and the serum amyloid A levels has been increased within the CAR-T treated group, and which indicating the liver damages associated with the cytokine release syndrome. They also demonstrated that if you put in the B-cell lymphoma or the myeloid cell lymphoma, the CAR-T can specifically targeting the tumor cells and dramatically increase the survival of the animals. But more than that, they use the humanized platform to evaluate the treatment to mitigate a cytokine release syndrome. So they first pre-treated mice with tocilizumab or the anakinra. The tocilizumab is anti-interleukin-6 receptor antagonists, it is used clinically to treat CRS. And anakinra is anti-receptor 1 antagonist, and it is also used clinically for the macrophage activation syndrome. So what you see here from the results is that the pretreatment with either tocilizumab or the anakinra that's not affecting the tumor clearance. If you compare to the red line or the blue line, they were pretreated with either tocilizumab or anakinra. And again, the pretreatment can also increase the mouse survival, especially the anakinra has shown a very strong effect in improving the survival of the animals. And more importantly, the anakinra can reduce the meningeal thickening, and which is an indicator for the neurotoxicity caused by the cytokine release. Another example of the Oncology platform application is the CAR macrophages. And in this particular study, the researchers transduced this anti-HER2 construct into the AD5F35 vector under the control of the CMV promoter. So one highlight is the CD3 zeta intracellular portion of the construct has a lot of homology with FC common gamma chain, and it can initiate the phagocytic responses similar to antibody-dependent cellular phagocytosis. And they also found no matter of the encoded transgenes is a vector or is the anti-HER2 sequence, that this vector itself can induce the pro-inflammatory macrophage phenotypes. And also the CAR2 target specifically in vitro can resist the phagocytosis and the lysis of the SKOV human ovarian cancer cell lines, which are positive for HER2. And then they tested the in vivo efficacy by IP injection of the SKOV tumor cells into the NSG-SGM3 mice. They treated the mice with either control, untransduced macrophages, or anti-HER2 CAR macrophages. And you can see that the anti-HER2 CAR macrophages can suppress the tumor growth for majority of the tumors if you check the red lines for individual tumors. And again, it improves the survival of the animals for over 100 days, which is the study date. And the next, they evaluated the bidirectional reaction of the CAR macrophage within the human tumor microenvironment in the humanized immune system model mice. So they used the humanized model by transplanting the female hematopoietic stem cell into the NSG mice, and after confirmation of the human myeloid cells in the peripheral blood, they engrafted the SKOV3 ovarian tumors subcutaneously and injected the male untransducted the macrophages or the CAR macrophages in intratumorally after 19 days. And lastly, they harvested the tumor and they removed the transferred macrophages by excluding all the human male cells, and then they did single cell sequencing, and then they clustered the tumor microenvironment actually into two different clusters, the cluster 0 and cluster 1. And the cluster 0 was enriched in the CAR macrophage treated arm, which has a high enrichment of the pro-inflammatory genes such as the MHC2, the TNF, and which demonstrating the CAR macrophage can remodel the tumor microenvironment towards the pro-inflammatory status. And cluster 1 is actually the more you have more untransduced macrophages group. So the summary for chapter 3 is that the human immune engrafted mice are unable to co-engraft either human tumor cell line derived xenograft or the patient derived xenografts. The human tumor immune cell infiltration is controlled by tumor intrinsic factors. Engrafted human immune cell populations are responsive to monoclonal antibody therapies that can up or down regulate the cellular responses. And the human targeted specific, clinically approved, and experimental monoclonal antibody can be evaluated for efficacy and mechanism of action. And lastly, the human immune engrafted mice are highly modular platforms that are capable of interrogating the monoclonal antibodies, bispecifics, and chimeric antigen receptor preclinical efficacy. So in this last chapter, we'll also share one application of the CD34 hematopoietic stem cell humanized mice for autoimmunity study. Again, you have to humanize mice first and by injection of the Priston, and you can also you can induce the lupus phenotypes, and then you can induce your therapies to see how it works in the humanized mice. And in this particular report, these researchers in the study has actually tested the CD34 humanized mice and then re-inject the Priston into the humanized mice. And they found by only a single injection of Priston into the humanized mice, it can recapitulate the key systematic lupus erythematosus features, which including the human anti-nuclear and onto antibodies expression levels, as well as the lupus nephritis that has been characterized by the immune deposits in the glomeruli, as well as you can see here by the IHC stainings. You can also observe the lymphopenia, which is a common feature that one of diagnostic criteria for SLE, but it's not often recapitulated by the SLE animal models. And you can observe the hyperactivated B cells and T cells, the elevated human pro-inflammatory cytokine. Proteinuria can only be observed in the Priston-treated humanized mice, and splenomegaly has also been observed in the Priston-treated mice versus control. So the application of the Priston-induced humanized mice model is illustrated in the following publication. In this study, the researchers actually trying to explore a new therapy that using the CAR nature killer cells to targeting the high PD-1 expressing human follicular T cells for the autoimmune disease therapy. And the rationale beyond this study was because the aberrant follicular T cell responses are associated with the development and persistence of pathogenic autoantibody secreting cells in the multiple autoimmune disease, while the PD-1 is a selective marker of the human follicular T cells. So they studied to using the CAR-NK to target these follicular T cells. And from the in vitro assay, they actually generated the co-culture of the follicular T cells with B cells in a different ratio. And then they do find the CAR-NK can specifically targeting the follicular T cells, as shown here by the increased levels of the lysis. As well as you can see the percentage of PI staining showed here. And lastly, it can also reduce much lower, less supernatant IgG in the wells that containing the CD4 positive T cells pre-cultured with CAR-NK. So I'll wrap up very quickly. For in vivo studies, they do find that the CAR-NK can specifically deplete the human the PD-1 expression in the by the CD4 positive T cells, as well as they showing the decrease the spleen weight once treated with CAR-NK compared with the control. A very brief summary for this chapter that the human immune engrafted mice treated with Priston can mimic disease phenotype of human systematic lupus erythematosus. The human T follicular helper T cells are icosal and PD-1 high, and it can support the humoral responses. And lastly, the CAR-NK treatment can reduce the splenomegaly and lower the serum IgG levels. So the conclusion for today's talk is that we have a robust platform of the NSG-based genetic derivatives, and which are capable of robust human multi-lineage immune reconstitutions. And the human immune system engrafted with mice can co-engraft with wide range of human tumors, and it is not HLA dependent. Also, the tumor intrinsic factors can control the immune cell infiltration. And lastly, we have the humanized platform and services to enable the preclinical evaluation of the human targets, specific immunomodulatory therapies, including monoclonal antibodies, bispecifics, and different CAR-T, CAR-NK, and CAR monocytes. The last slide I want to share with you is that Jackson Laboratory is here to provide solutions, to provide you with more the different models or through model generations, and we can also provide services for the targeted validation and the preclinical solutions for different research areas.